From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on November 28th, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 117th episode of Matters Microbial. Thank you for every view or listen, and please spread the good microbial word. It has been a bit since a new episode of the podcast, for which I do apologize. We're trying to get into a new mode with my looming retirement and shuffling schedules. Thank you all for your patience. This podcast means a lot to me, and I hope you enjoy it too. As my last course in microbiology at the University of Puget Sound steams ahead toward its conclusion, my Micronauts in class are thinking about the entire course. First, this quote from former podcast guest Dr. Mark Chevret says it all. Every so-called rule in microbiology seems to have an exception. Some students find it frustrating, but there is literally something new to learn every week and to marvel at. This quote from a former colleague of mine, Dr. William Metcalf, is a reminder that what we test in the lab is for our purposes, not for the purposes of the microbes. It's easy to forget. Micronaut Kaya continues to take her tardigrade to swim meets seen here. I do try to promote a microbial point of view regardless. My Micronauts created many memes about microbiology this semester. First is a nice one from Micronaut Jonah, seen here, reminding us that eukaryocentrism is a trap into which all biologists can fall. Micronaut Ava was very taken, as seen here, with the idea of microbes in her drinking water. Her water bottle buddies, as we call them. This intersection of art, creativity, and microbiology fascinates me. And Micronaut Kylie modified a Katy Perry song into I Kissed Agar, as seen here. I feel like hiding from the pun. Microbial humor is everywhere, even with a darker tone, as seen in this bit of art made by a former Micronaut which hangs on my laboratory wall. What doesn't kill you mutates and tries again. Eek. Micronauts continue to find the most interesting microbial denizens on their plates, and observation is key. For example, what are these donut bacteria seen here, found in a marine sample? Are they mucoid colonies that then collapse? Well, they all certainly do. Or these circular patches on a water bottle swab seen here. They are not apparently phage plaques, but most likely gliding microbes. Most of all, I love to share the beauty of microbiology, as seen in this photobacterium dilution plate seen here. It really does look like a starscape to me. Beauty. In my many years of teaching biology, I have worked very hard to show the human side of research and even how unlikely research becomes important. That is, how did Messelson and Stahl know about the semi-conservative model of DNA replication? That sort of thing. So, the history of biology is important to me. And it turns out that several types of viruses that can in fact cause cancer have in turn taught us a great deal, providing enzymes and approaches central to modern molecular biology. So it's a distinct pleasure to introduce Dr. Gregory Morgan of Stevens Institute of Technology to the podcast to talk about his work on the history of biology and in particular his fascinating book, Cancer Virus Hunters. Welcome to the Quality Quorum today, Greg. It's great to meet you.
Thank you very much, Mark. I'm very happy to be here.
I noticed that you have training in molecular biology, but you've become a science historian. And I wanted to tell listeners and viewers that this is just speaking to my center, because I was so influenced by the book, The Eighth Day of Creation, and I'll put a link to that book in the show notes, and of course, a link to your book as well. What first interested you in the history of biology?
Well, I grew up in rural New Zealand, and on a sheep farm, which is a little bit it's a little bit passé perhaps for coming from New Zealand, but and we were interested in things like the genetics of wool producing sheep and the genetics of velvet producing red deer. And so I was interested in biology, I think, from the beginning. And then so I studied at University of Otago in New Zealand, and then became an exchange student at University of California, Berkeley. And I ended up as a biochemistry major and an economics major, but I really thought they weren't speaking to me in the sense that I could see a whole career in those. So I was actually wandering around the United States in 1991 because I didn't have a work permit. So I just traveled around the US, and I went to Powell's bookstore in Oregon, and I picked up a book on the philosophy of science by a guy called Rudolf Carnap. And I thought, oh, this is amazing. This is people that are interested in science, but they're not actually working in a lab. They're interested in the conceptual issues. So then I discovered that there was a field called history and philosophy of science. And so I decided to apply for graduate school there. And there's a big center at the University of Pittsburgh. It's very strong. So I ended up going there for graduate school.
That sounds really interesting. I mean, I'm older than dirt, so some of the people that did some of the work that are central to what I teach, I knew them. And so that's like very bizarre to me. I was just telling students about meeting with Paul Boyer and taking biochemistry from him when Peter Mitchell got the Nobel Prize, and
Oh, wow.
how surreal that was. He
Right.
was not happy about that, though, but that's another story. And again, it comes back to the fact that honestly, and it's really important to me, science is a human profession, and people seem to forget that. I remember my late mother saying when a very famous scientist had been in the newspaper and said something that was intemperate, so I put that carefully, and my mother turns to me and says, "I thought scientists were supposed to be smart." And then I said, "You don't know many scientists, Mom, but it's the truth. They're like anybody else, and there are ones that are thoughtful and ones that aren't. It's a human profession."
Right. Yeah, I think for me, I didn't really understand that until graduate school. I feel like the undergraduate biology curriculum kind of wipes a lot of the history out of the textbook. And so you get the results of the experiments, but you don't learn much about who the people were that did them and why they ended up doing the experiments they did.
I certainly talk about, some of the sociological aspects of it. It's easy to talk about Rosalind Franklin, for example, but it's a more nuanced story than we often see shown, is the interesting part
Right.
to me. And that's, I think, fascinating. But a lot of times students would say to me, "Why do I have to learn about the original transformation experiments?" And I'll say to them, nobody knew what the genetic material truly was.
Right. Yeah, Rosalind
And
Franklin is someone that I'm quite interested in,
ah,
partly because she was doing work on viruses. People think of her in terms of DNA, obviously, but Crick, Watson, and Rosalind Franklin actually were interested in viruses and doing experiments on them in the early 1950s,
yeah.
mostly through X-ray crystallography. So they weren't as interested in the genetics as we are maybe today, but they were interested in structure, biological structure, and viruses were a good way of getting at that.
I couldn't agree more. And it's as I say, I think I don't know who said it. Maybe you do, since you are. Can I call you officially a philosopher? Because I believe I can.
You can, I guess, I'll accept that. Philosopher
So
of science. Yeah.
I don't know who said this, but I love the idea that history doesn't repeat itself, but it does rhyme.
Right.
And it's true. I don't know. Do you know who said that? Because I don't.
I definitely have heard it before. I can't attribute it right off the top of my head.
Okay.
Yeah.
That
Sorry about
happens to me a lot. So I think the right thing to do now is that we're focusing a little bit about your book, which I have.
Right.
And you have, of course. And the whole idea is the idea that is the hook is there are some viruses that in fact cause cancer, and how studying these types of viruses as a human profession have actually informed modern molecular biology. Do you have a place you'd like to start?
I guess a good place to start would be kind of at the beginning of the book, which is Peyton Rouse, um, who's a scientist, pathologist working in New York City. He's very much interested in very careful observation. And one day in 1908, I think it was, someone brought in a chicken with a cancer in its breast. And so he was interested in the nature of cancer. Very little was really understood about cancer at this time. But what he was able to discover was if he took the tumor and he chopped it up and ground it up and then filtered it and injected what he had left over, he could create another tumor in another chicken. So it looked like cancer was sort of contagious in this way. And this is really the beginning of what we might call tumor virology, the idea that some cancers can be caused by a virus. Rouse didn't know it was a virus. He didn't call it a virus. He called it a factor or an agent. And virology really is just in its very beginnings at this time too. We can't see viruses. We don't have electron microscopes that show them. We just know them as things that can get through very, very fine filters, so they must be smaller than cells.
I think there was a Sherlock Holmes story about this, in the sense that they didn't, and they're clearly talking about a virus, but they didn't call it a virus. They called it anyway. The point is, you're exactly right. The idea that it is something that is so small it's hard to believe that a cell could be responsible for this. Though there are a few examples of contagious cancers which fascinate the heck out of me.
Right. Yeah. I think it was even up for grabs if it was a biological thing or a chemical thing. You may be aware that you can crystallize viruses. So the fact that they can be crystallized makes them seem like they should be chemical rather than biological. Yeah. So it's sort of an interesting period of flux at the beginning because virology isn't very well formed. Neither is cancer research. So we don't really know about the nature of cancer or about viruses. But Rouse thought he was onto something pretty special. Other people were more skeptical, wondering maybe this is just a sort of an interesting tweak of chicken biology. It didn't apply to anything else. But Rouse did publish on this and got the word out that this was a possibility, that there was something biological that could cause tumors. In some sense, his research program ran out of steam because he didn't have the equipment that we would use today. In particular, he would need a centrifuge or something like that to purify the agent. But then other people picked this up. There was a guy called Shope, Dick Shope, who discovered that something similar was happening in rabbits. You might have heard of the legend of the jackalope. Have you heard of that? Where rabbits have these big horns that grow out of their heads. That's caused by a papillomavirus. So Shope did something similar, where you would purify the biological growth and then filter it and look at what goes through the filter and inject that into new rabbits. He did this at Princeton, and then a third guy did something similar with mice. And so now we're getting into like the late 40s, early 50s, where it looks like a number of different animals can have cancer-like growths caused by some very tiny biological agent. It's not really until the 50s actually we get our modern conception of virus sort of comes into focus, where we see it as a little piece of nucleic acid surrounded by a protein coat.
Well, it's interesting. It's interesting you say that, because for every little piece of art that we have,
right,
even a supposed polyoma virus, I tell students that, work prior to the early 50s, they weren't really sure what the genetic principle truthfully was. They thought it should have been DNA and it should have been, but they didn't see how. And that's because they didn't think about reading the code in a linear fashion.
Right.
But at the same time, if you look, and you mentioned this before with a very simple virus, tobacco mosaic virus,
Right.
RNA, single type of protein, self-assembles, and you can crystallize that thing. I have
Right.
seen it in bottles, crystallized,
Right.
and then you abrade a tobacco leaf and rub some of that in, and you start to get the blighting from that. So the idea of very small transmissible agents are one thing, and we're used to the idea of them causing types of disease. But what fascinates me about your book is how this was the crowbar that opened up a little bit about how our own cells work.
Right.
And that's, to me, the really interesting part. I'm delighted to hear you pronounce Peyton's last name, because I was never sure, and now I'm petrified to say it. How do you say it?
Rouse, I guess.
Rouse? Okay.
Rouse, yeah. I have a New Zealand accent too, though, so that's another layer, perhaps.
Well, for me, I'm old enough that there wasn't a way that you could click on something to know how to pronounce it. So I get overly enthusiastic, so I was teased a lot about this growing up. But the RSV, the Rouse sarcoma virus, is just a fascinating example of the whole thing. But he didn't know anything about it. But I believe he got the Nobel Prize. Was he the oldest person to get the Nobel?
Well, I think if he's not the oldest, it's the biggest time period between when he did his research and when he got the prize. I think it was 1966.
Yeah.
So he was an old man at this point in time. He'd done the research in 1909, 1910. Yeah, so it was sort of one reason it was the big delay, of course, is that a lot of people were skeptical that viruses could cause cancer. There was other theories. Obviously, we learned that tobacco was carcinogenic, or smoking tobacco at least. And there were other people that thought that maybe the chemical mutagens.
Yes.
And now we have a more complete picture, and all these things can play a role. But at the time, they looked like competing theories. Some of the early people thought that all cancer was going to turn out to be virally caused, but it really is only roughly 20%, I think, is the estimate people give today. But yeah, you're right. It was the kind of key to unlock the molecular basis for cancer. And I think there's a number of reasons why that was the case.
Well, I love the idea of trying, and again, I want to emphasize to listeners and viewers, if you can't see it, it's kind of hard to describe it. And that sounds like a weird thing to say, because certainly chemists aren't seeing molecules, but they can describe how those molecules interact.
Right.
So in a similar way, when you have a good example of a virus that can be crystallized, like tobacco mosaic virus, it's very easy to think of this more as kind of a chemical sort of thing than a biological entity, sort of.
Yeah.
You even wrote a paper, "Are Viruses Alive?" What a hero you are to me.
Right. Yeah, it's interesting because you mentioned TMV, it doesn't actually form true crystals in the sense that it's three-dimensionally crystalline, which make it more difficult to analyze. And you mentioned Rosalind Franklin was actually looking at TMV in the beginning. But some of the plant biologists at the time weren't even sure if it was correct to say that every virus was exactly the same size.
Yes.
So she was sort of fighting that battle at the time to say, no, we can imagine them that are all little cylinders stacked on top of one another to make up a crystal.
And electron micro. Go ahead.
One reason people were interested in TMV from a more theoretical point of view was that it was one way of getting at the structure of RNA, right? Because the thought was if you could work out the structure of the whole virus, then you could subtract the protein part and work out what the RNA, what the nucleic acid was doing inside the virus. And Don Caspar, who was one of Rosalind Franklin's friends, worked on this with her. And you could take electron micrographs of the virus and also of just the protein part of the virus, and then compare to see where the RNA was. And it actually turned out that Watson and Crick came up with a model of RNA and TMV in the early 50s. It was never published, but it was another helical structure. The idea was there was lots of RNA chains that were helically arranged inside this cylindrical um, TMV virus particle.
But by then, a little bit of electron microscopy had started. I want to say 1946, 1947. Am I wrong?
That's about right. Yeah, I think
Mm-hmm.
the big advance was the invention of negative staining,
Right.
which allowed you to sort of see things. So a lot of this happened at the University of Cambridge in England. Sidney Brenner was involved in that. You probably heard
Yes.
of. But the idea that if you could have a very heavy metal surrounding whatever you want to look at, you kind of get an image that almost looks like a photograph. It's not quite a photograph, right? Because it's a projection through the material, but you could see viruses, and eventually in the 50s, you could see those subunits of viruses. So you could see that they were made up of something. So then the question was, what are they made up? How are they arranged? And this is what Watson and Crick were interested in.
Mm.
Um, they were able to, I think it's the first example of an information theoretic argument that they gave. So they worked out how much nucleic acid was in a particular virus. That was Bushy's stunt virus, actually. And then they could work how many proteins roughly would that code for. And it was a small number. So then they confer that there must be a small number of proteins that make up the rest of the virus that's repeated in some sort of regular way. And they published this in 1956.
Mm-hmm.
It's not it's sort of interesting because we all think of them as, we think of 1953 as the big year with DNA, but it really took another decade or so before most biologists realized how important this discovery was. And so the late 50s is an interesting period for molecular biology. You've got genetic schools and you've got people doing X-ray crystallography, and they haven't quite merged into a new discipline yet. It's just sort of beginning to merge into what we would call molecular biology.
I'm not going to throw Thomas Kuhn at you at all, but I am going to say, right. See, that your reaction is perfect to that. But it is interesting that we tend to build on things that we've already accepted as being true. And so it needs to have its time.
Yeah, I
And,
think that's
and so
right.
and so, for example, I don't recall the name of the German geneticist who was first to see bacteriophages, and perhaps this is apocryphal, but he supposedly said, "Mein Gott, they've got tails."
Right, right.
I don't know if that really happened, but it shows an important concept regardless, is once they had an image that they could start to think through. And
Right,
that's the value of model building, to come back to what you're discussing there. But does the model reflect what actually exists?
right.
So this brought together, as you say, a new field. And it really emphasizes one further thing, that I think that siloing of disciplines is dangerous. I always have, because you get in a giant, I'm going to change my language, a giant ring of people patting each other on the back saying, hey, you're very smart. But that's not really how science advances. It's when you come in and new ideas and you engage with them. Some of them are right, some of them are wrong, but everyone benefits from that. And I think a lot of the public doesn't understand that that's how science actually advances. So to bring in somebody who is, say, an engineer to help with building an electron microscope is very important.
Right. Yeah. I mean, in my book, I call one of the insights that I got from the century of research was that I called it methodological pluralism, but the idea that you had people generating information and data from very different points of view, but it was still useful to other people. So you had some people that were sort of Popperian falsificationists. You had other people that see themselves almost like field biologists out in the
Mm-hmm.
field. Um, and it all made sort of congeals into this field of tumor virology, which today is not that well known, but it really was the center at some point of molecular biology in the 1960s and 1970s. It's where, because viruses are small enough that you have a small piece of DNA or RNA, then it's only a small number of genes that you're dealing with. Before you had the recombinant techniques to make smaller pieces of DNA, you'd have to look at what a virus did. So it was like a little package of genetic information that you could build experiments around. So Kuhn, I think, as you bring up, like wants to think of things as having this big paradigm shift where everything kind of switches. Whereas I see more of it. Sometimes people use the word bricolage. There's little changes all over the place, and they all build together. Some things are rejected eventually, but I sort of a coalescence of a body of knowledge and a field of people that are willing to talk to one another. In this respect, Cold Spring Harbor Laboratory is a really important place because it gives a location for people to have conferences to come and speak from all around the world. And it really starts driving the field forward. I think Watson, unfortunately died recently and is remembered as for a number of different things. But one thing I think he's under-remembered by is his ability to actually build a field by showing that you need to have, yearly conferences and then dissemination of the results, which were in these red books that came out of Cold Spring Harbor that would have been then distributed to all the academic libraries in the world.
No, I think that's exactly right. You look at the kinds of work that came out of Cold Spring Harbor, it isn't just about, one type of field, but you bring all these folks together to talk about it. I remember Barbara McClintock's work was published in some of those quarterly reviews.
Right.
And I would listen to a member of my PhD committee, Alan Campbell, who said, and I said, no one understood Barbara's work. And she said, well, we understood it. We just didn't think it was important. And maybe he understood it because he is without doubt the brightest person I've ever met. But it's like reading a Letterberg paper. They're really complicated.
Right. Yeah. And McClintock ended up staying at Cold Spring Harbor for a number of years. They had a core number of scientists working there that would attract others to come and work. Nathaniel Comfort's written a really nice book about McClintock, which I'd recommend. This actually does tie up to a theme in my book too, which is it was difficult for a woman molecular biologist in the 20th century, and she's like one example of it. But there's a number of other examples in my book where the women were doing really good work, but they didn't really get the same level of credit as the men.
Agreed.
Yeah.
And this is something that our current society is starting to understand. If you do the work, you should get the credit, of course. And one of the things that you alluded to is how syncretic a lot of the work actually is, where you have little bits and pieces that can be brought together. And this is the value of proper communication. And if you get your idea of how science progresses from the race for the double helix, and I don't wish to speak ill of anyone at all, but I think that's a little bit sensationalist, or a whole lot sensationalist, depending on one's point of view. What a really important point of that book to me is showing it came from lots of different people doing different things that were brought together. And, we can argue about who should get more or less credit, but they were all necessary is the point that I took away.
Right. Yeah. I think that's a good point. It turns out that in the history of tumor virology, there was 7 Nobel Prizes given out, which is a lot. If you think about, it's a pretty good percentage of all the Nobel Prizes in physiology and medicine. In a number of cases, it's really difficult to work out who to give it to because there's a limit of 3 per prize, and there's more than 3 people involved. And this is one reason I think, like, for example, Mies Chow, who was an electron microscopist, who worked with on RNA splicing, didn't get the prize, whereas Rich Roberts and Phil Sharp did. And so you're right that you need. I think this increasingly becomes true as you progress in the 20th century too. In the early days, it was easier to be a solitary scientist who made important discoveries, but by the end of the 20th century, you really have to be a member of a team, and you have to have your team connected to other teams of scientists to be successful. So success is almost a social challenge as much as it is a scientific one.
I agree. And we can make jokes about it. I don't know if you've heard 3 stages of scientific research. The first is that's a dumb idea. You shouldn't have suggested it. The second level is, well, it might work in this particular case, but it's not relevant to the natural world. And the third is, I said it was a good idea all along.
Right, right.
And a student of mine came up with a fourth one, and I came up with the idea first. We've all seen this.
Right.
And it's that battle between people who want to take credit for things, looking out for themselves, versus the essentially social nature of how science actually progresses.
Right.
And
Yeah.
and those two dynamics are at odds.
Yeah, it's a balance between cooperation and competition, right? So you need to have collaborators and other people to bounce ideas off, but you also want to be first, right, and not be scooped by somebody else. So that tension, I think, drives a lot of the discoveries in the 20th century. Watson, in fact, made it more competitive than average. I think he wanted his scientists working at Cold Spring Harbor to be in competition with one another and with other teams around the world. And maybe that drove things forward faster than it would have been if it was purely cooperative. But yeah, it's the balance that's the key, right? How to balance those two things.
So to kind of move back to the tumor virology aspect, you were talking about Rous sarcoma virus, RSV, and no one really knew at the time what the virus was carrying that could cause cancer. And then folks discovered, if you'll forgive me, that there was a hitchhiker in that virus that isn't really necessary for viral life.
Right. Yeah, this and some, I think, is some of the most important molecular biology of the 1970s, right? When we are now able to get tools to look at the genomes within RSV. And there was a number of labs. There's an important lab, a number of on the West Coast where you are actually. But at UCSF, we had Varmus and Bishop,
Yes.
who had a very important collaboration. They were looking at this intently. There was others further south in California and up north in Washington that were looking at similar problems. And they formed a kind of cooperative where they were looking at this. And they were able to show, just as you mentioned, that, yeah, there's one piece of the genome which had the oncogenic potential, and there could be viruses that didn't have that little piece. So those two facts, when you sort of put them together, you realize, oh, so the gene, the oncogene, as we would call it today, isn't actually viral in its beginning. It's that the virus has picked up the gene from something else, maybe a ancestral chicken, and now it's able to create transform cells or cancerous cells when infecting hosts.
To me, it's really interesting because when I would teach this subject, and the idea of a gene that you say can promote, I'm not going to say cause, but promote cancer, we call that an oncogene. And it's usually involved in cell signaling or telling a cell to cease or to start to replicate.
Right.
That's very simplistic, but those are the ideas. And then you think, why on earth would a virus pick up a hitchhiker like that? Number one. But number two, if you're studying an oncogene and you discover that it has a perfectly normal version that you need in you, what do you call that? And they came up with the clunky term, and I understand where it comes from, proto-oncogene.
Right.
And this really confuses students. It's right up there with why does photosystem 2 come before photosystem 1 in the Z diagram. And I always tell people that it's I didn't discover it, and it's what's discovered first. So one thing that really struck me about this concept is something that I've introduced to my students. It doesn't have to be about something that promotes cancer. For example, blue eggs are due to a retrovirus, and you'll talk about those, I presume, in a moment, that's integrated near and overexpresses a little pigment gene, and now it's inherited like part of the genome, because it is. part
Right.
of the genome now. And even the issue of placenta.
Right.
Right. And so
Yeah.
what this has been the blooming of this idea, because the original question is it just by accident that you pick up these genes that can cause cancer and express them inappropriately, which is the key? Or is there something general going on? But once again, just as I said with Bill Metcalf, the way that we study things prejudices what we find. And so if we're looking at things that cause cancer, well, there you go. You're going to be looking at things that cause cancer. And what I'm trying to say earlier is that it looks like a more general thing. It might be involved with genome evolution, in fact.
Right. Yeah. Yeah. I mean, this one of my guiding research principles has been that in 20th century biology, at least, the viruses are actually the most important organism that drive so many discoveries forward. And you're just mentioning something now, which is more recent, right? That maybe a lot of even human evolution or eukaryotic evolution is driven by viruses using horizontal gene transfer or getting stuck in the genomes of their hosts is actually really important from an evolutionary point of view. I'm not sure if there's a consensus about whether or not oncogenes make viruses somehow fitter than ones that don't, or that it's just sort of luck. Presumably, the RSV is picking up a whole bunch of different genes, and we only notice the ones that cause tumors, right? So, I guess it's a mismatch between how much DNA or RNA you can package in your virus particle and how much you need to replicate. And if you've got spare room, you can pick up other things. In theory, you pick up things that are useful for the viruses in the long term. But I'm not sure. I didn't take a stand on this in my book whether or not that was selection pressure for picking up oncogenes as opposed to just sort of a lucky event.
Well, at the risk of sounding eukaryocentric, which is one of my deadly centrisms, the amount of rDNA that actually encodes for proteins is tiny. And about 18% of our genome is made of what's called an ALU sequence, which is clearly a remnant of an RNA virus, and it's 18% of our genome. So the idea of viruses interacting in the eukaryotic genomes for sure,
Right.
with how genomes get larger or smaller. And then, as you say, if you're looking for something, you can kind of find viruses associated with that, but it doesn't tell you what the generality is. Just
Yeah.
like with transducing bacteriophages, it's the same principle.
Right. Yeah. I think if you look at the history of virology, there is, at least by the 1960s, the field actually divulges into two different research programs, depending on what type of virus you're looking at. There was some that were looking at DNA tumor viruses and some that were looking at RNA tumor viruses. Um, RNA looked promising because of another discovery in the 1960s called feline leukemia virus,
Yes.
where cats could get leukemia from an RNA. So the federal government actually spent hundreds of millions of dollars trying to look for a human equivalent, thinking that maybe human leukemia was caused by a virus. And a lot of that money actually some of it went to Cold Spring Harbor, but a lot of it funded basic virology research in the 1970s as part of Nixon's war on cancer.
Yes.
And so we didn't find this leukemia virus that causes lots of human leukemia, but we did a lot of really other good work, including the RNA tumor virus field and the DNA tumor virus field. And so, as you mentioned, the oncogene discovery was a discovery first with a gene called SARC, which is a kinase, and then second with another gene that everyone's probably heard of if you study molecular biology, called RAS. But they actually combined in some sense with discoveries from the DNA tumor world, where P53 was discovered as a host protein that was interacting with virus proteins when they infected a cell. And that's a really, really important, as it's tumor repressor protein. And together, it gives us a more complete picture of what cancer actually is.
Mhm.
I think Vogelstein, Bert Vogelstein from Hopkins, had a nice analogy where he said, well, to make a cancer cell, you have to destroy or you have to destroy the accelerator of the cell so it gets switched on, and then destroy the brake as well so it gets switched off. And so these two different types of switches map onto these two different types of viruses that people studied.
It's funny that the National Cancer Institute uses that particular metaphor of a race car
Right.
in some of the visuals.
Yeah, it's a good one for people. You can imagine it's more complicated than that. Obviously, there's probably a lot more switches than one accelerator and one brake. But what makes some of the research harder to interpret looking back is that we're using cell lines that already had certain damage to them already. So when
Yes.
you infect them with a virus, it could do things like cause cancerous cells that it might not do when you give it to normal cells in an organism, say.
It it's really common when I talk to people, and I'm thinking of my late parents, that don't have any experience, and they're very smart people, but they don't learn a lot about, they haven't learned a lot about science or how science works, or they get it off, Lord help us, television. Nowadays, social media, even more frightening than television, right?
Right.
Except for this podcast, of course, and the other
Oh,
members of Microbe TV. But what I'm trying to get at here is that someone could look at this work, say, with feline leukemia viruses, and they're going to kind of give you like, why do you care about that? But you can at least say it's related to this issue involving leukemia in people. Maybe, and we need to find out. And people can accept that kind of idea.
right.
And when you go from that to something like, why do you care about cancer causing viruses per se? What we've learned about this is it's taught us a lot about what controls cell division and proliferation in us normally. So
Right.
to my mind, what started, and I know this isn't exactly accurate, please forgive me, it sounds almost like it's applied, you're looking at cancer issues, but you're actually looking far more basically. And I think that when you find out, for example, there are relatives of proto-oncogenes in yeast, that tells you what you need to know right there. We're at the very basics of the blueprint of life that we need to understand if we want to make the changes that increase the quality of our lives.
Right. Yeah, I think one theme after looking at a century of research is that some of these breakthroughs take a lot of time and sustained effort. And I wonder sometimes if these short-term grants that we give out now are kind of antithetical to these long-term research programs over decades.
Mm.
Um, but yeah, a lot of our advances in understanding cancer and the normal cell cycle, it doesn't even looking at human cells, right? So cell lines from different animal species have been really important. Veterinary science has a role to play. Um, and this idea that sometimes you let these really smart people just investigate what they think is interesting with the hope of some sort of unplanned payoff, it has actually worked out quite well.
Mm.
Um, one of my favorite stories in the book is actually that by looking at RNA tumor viruses, we discovered reverse transcriptase, which is a really important enzyme because it allows you to detect RNA. Um, and that seems sort of esoteric for people that aren't biologists. But then when the HIV pandemic happens, it turns out that's a retrovirus too. We actually end up renaming RNA tumor viruses retroviruses in the 1970s. We had a test for HIV really, really quickly because we'd done this earlier research. And we'd be in quite a different position if we hadn't done this earlier research. So people like Robert Gallo, Montagnier had done a lot of this basic research that then could be more easily applied. Um, yeah, I think that that's a theme that you see in 20th century biology too. That it's you can't jump straight to the applications. You really do need some of the basic research done first, which could take decades in some cases.
A lot of people are into that, and one of the goals that I want to have if I promote science at all is that the process of science needs to be better understood by the public. And it's very easy to have, and I'm not being at all partisan when I say that, because every politician does this, where they point fingers at what they consider to be wasteful research.
Right.
And I can't make a judgment whether that's true or false. What I know is that what we're calling basic research can have a huge impact in unanticipated areas.
Right. Yeah, it's a hard sell if you're a politician that's on a 2-year cycle, or right. You
Just
want
like
to,
grants. Just
yeah,
like grants, right?
right. Yeah, it does seem like there's a lot of waste of time in that, trying to justify your research in advance, as opposed to having more open-ended grants that are just given to young investigators to sort of do what they want with. I guess that's sometimes called blue skies research, but
Yeah. Yeah. Well,
uh,
the Howard Temin story is a fascinating one to me, and I'm glad you included it in your book.
yeah, well, Temin is one of the heroes of the book, I think, of anyone, because uh, there was a puzzle, of course, if you have RNA tumor viruses, and how do they replicate? Because the central dogma of molecular biology, as Crick and Watson kind of describe it, was the idea that you couldn't get information from RNA to DNA. And Temin just thought, well, there must be a way, right? It
Mm-hmm.
must happen. Biology must find a way. Most people thought he was misguided. So for years, he was, I think, derided,
Yeah, he was.
until the discovery of reverse transcriptase, that he discovered, and in Baltimore, David Baltimore at the same time discovered it, and then he's vindicated, um, and seen as a very insightful guy. I enjoyed, he had a long discussion with Francis Crick about his discovery, because and that's in the archives at the University of Wisconsin. Um, Crick actually was more open to the idea that you could get information from RNA to DNA, um, and that it depended on really how you interpreted the central dogma, how strict you thought it was. Yeah, and so Temin, I think, is a really important figure. He's obviously presenting at Cold Spring Harbor too, and there were other people that kicked themselves because they thought they were looking for reverse transcriptase. But it's kind of amazing in some ways that these viruses will have the protein inside their little particles ready to go. So
Yeah.
as soon as they infect a new cell, they can take their RNA and turn it into DNA. Um,
So when I teach students about this a little bit, and I talk about positive and negative single-stranded RNA viruses, and how a particular type has to pack their own lunch, meaning they have to carry the enzyme with them, they get very frustrated and say, well, why can't they just be positive strand viruses? And I quote my virology professor, Arnie Burke, when I asked a similar question all these years ago, and he said to me, that is a theological question, which I've always enjoyed, right?
yeah,
Because sometimes that's just what it is. And I say this and it upsets evolutionary biologists. I'm not sure we understand selection as well as we think we do. And I think fitness is we call fitness something that we just lump a bunch of stuff into. And that's not how it works, I don't think. So I'm not trying to be vitalist and saying if something exists, it has to promote fitness. That's not what I'm saying, because
right.
everything is stochastic. But I think that we miss a lot, and we need to have the time to think. And this is something that you've said several times in your book and today. In some ways, we need to have more time to think
Right. Yeah.
and really consider, because these things have been around more than 1 billion years,
Right.
right?
And actually, the thing that I think blows some people's minds is just the number of viruses that are out there too. That
Yes.
they're by far the most abundant biological agent on the planet, and they've presumably have explored kind of design space over those billion years and discovered all sorts of little hacks and ways of doing things that is
I
good
like
for.
the way you put that, exploring design space, because I do think I read a lot of science fiction, so maybe that infects my brain. But that is the point, isn't it? And you look at the effect that viruses have, and just like the original focus that early microbiologists had on bacteria that cause disease, we kind of do the same thing with viruses with very few exceptions.
Yeah.
And I really worry about that. What else are they doing? How would we find out, right?
Yeah. I mean, from a philosophical point of view, I think what it shows is that some sort of methodological reductionism is the way to go, right? So I know biologists don't want to say, well, we don't want to reduce our entire field to chemistry or physics. So it's not an ontological reductionism. But from a methodological point of view, focusing on a really small, simple system and understanding it really well has been the key to success and successful scientific careers as well. A lot of these guys that I study in my book, um, they have a post-scientific career in administration of some sort, right? Varmus becomes the head of the NIH. Tejan becomes the head of Howard Hughes Institute. Um, Bishop is the chancellor of UCSF. They all, and then there's people that go into the private sector too. So it was a very efficient way of having a successful career, and it paid off for a lot of these guys. And although, I mean, biology is complex, you look at your textbooks, there's so much details. It does seem like the way that we've made advances, at least in the past, is to simplify and try and understand a simple system first. I think I enjoyed your point earlier where it was a lot of physicists that came into the phage group in the 1940s and 50s that had that orientation as well. And it basically infects molecular biology and molecular biologists to think this way, I think.
Well, I mean, if Max Delbrück and his general philosophical followers, for lack of a better term, they wanted to be able to put a gene in a mortar and pestle. And so they had to figure something simple about it. And the idea of a virus that infects and attacks bacteria. And when you talk about now, I'm looking at whether or not the phage plaques are turbid or clear, large and small. And people can roll their eyes about that. But that was necessary work to create the framework that we built
Right.
on.
Yeah, and I think actually explicitly the switch from prokaryotic viruses to eukaryotic ones was explicitly made by trying to replicate what the phage group did. So you need an assay that's like the plaque
Yes.
assay for eukaryotics. And so that was done by Delbecco and others to work out how to grow a layer of eukaryotic cells that then you could infect with eukaryotic viruses. So, yeah, we emulated the success of Delbruck and Luria and Hershey and all those guys by importing their sort of approach to biology into the eukaryotic sphere.
Well, I mean, if you look at, for example, this 3D printed model of a T-even bacteriophage that I'm showing you
Yeah,
now,
looks like
um,
T7. Okay.
couple hundred proteins, they have to be assembled just so. And I know people in the early days that said, I want to study that so I can get into developmental biology. And people would say, well, that's just a bacterial virus. However, compare that to tobacco mosaic virus that can self-assemble,
Right.
right? So you have to start somewhere and build from it. And that's clearly what they did. And what I appreciate about the tumor virologists in particular is that starting from something of a medical interest, essentially, with a phenotype that's relatively straightforward to see, transformed cells, they were able to shed light on what takes place in every cell among the eukaryotes.
Yeah, that's right. So, I mean, one way of thinking about this, it seems to me, that's useful is the virus is a small number of genes. It's trying to attack and basically take over a cell. Uh, what host proteins it interacts with is really, really important, right? It's a bit like if we were taking over a country, we were like invaders. The people we interact with to do this are really important. So, in a way, the virus helps us out to find out which proteins, the host proteins, are really important. So, as we mentioned, the proto-oncogenes end up being really important genes for normal cell functioning, and same with genes like P53. Yeah, so it makes sense from a certain point of view that this is the way we should think. If you've got tens of thousands of host genes, which ones are the most important ones to study first? Well, not a bad heuristic to say it's those genes that have to be modified or changed or stopped by a virus when it wants to take over a cell.
Oh, certainly. I mean, we have, depending on who you talk to, between 25,000 and 35,000 genes in you and I. And I know that that's a flexible number and doesn't include any number of modifications on what we define. Nonetheless, that's a large enough number, it would be very difficult to study it. So to have a smaller subset from which you can study makes a difference. But technology improves things. For example, in my own little lab down the hall, I can have a known organism where all of the genes are mapped out, call them 4,000 in a bacterium. I can look at a mutant. I can submit that genome. And for about $75, I can know how that mutation has affected every gene. In other words, if there have been secondary changes or tertiary changes, I can figure those things out. I never would have dreamed of that before, which makes me think about the next 15, 20 years.
Right. Yeah, no, it's kind of amazing. I mean, I picked history and philosophy of biology partly because I love biology, but partly because it's one of the most important transformations in the 20th century, if you think about it. There's a revolution in physics and there's a revolution in computer science too, perhaps. But where we've gone from 1953 to now is unbelievable. And the way I like to describe it is we're moving from the agricultural phase of biology to an engineering phase, perhaps, where we're going to start engineering things more and more. And I actually end the book with this point where people take viruses to try to make them and modify them to try and attack cancer cells. So now instead of being a cause of cancer, they can maybe be a cure. And I see that increasingly starting to happen as we basically turn biology into a form of engineering.
I think that is the future. Freeman Dyson used to say that, and I always enjoyed his books on this subject about things because he came at biology as a physicist, and I think more of a mathematician, actually, but you get the idea. Right. So certainly it brings us full circle into what we learn from people outside the subfield in which we have toiled.
Right. Yeah. Yeah, I mean, one reason I wrote this book, and I tried to explicitly make it readable for scientists and the educated general public too, was this worry that it's sort of bad for our culture when we have a public who doesn't understand what scientists do, or maybe don't even know a scientist, right? I think it makes a big difference for people if you know someone, and like I say, I know Mark, he's a trustworthy guy. Maybe I should believe what he says, or at least consider it. But that means we need, I think, better science communication and trying to convey the beauty and majesty of scientific discovery to people that aren't scientists.
I have, and I think you'll appreciate this, and I know I'll make you laugh. But I have a friend who taught at a pretty important small liberal arts school I won't name. And what this friend of mine had is difficulty getting the administration, who were not biologists, to understand what it was like to do experiments. So by force of will, and if you knew this person, you would understand, my friend forced administrators to come to their lab and do an experiment. And then they understood what was necessary.
Right. Yeah.
And that's the key to bring it across. I'd like to frame something for many listeners and viewers that has been much in the public eye recently, and that's the issue of cancer and human papillomavirus, HPV. Would you mind going over that a little bit? Sure.
If I was to pick out one of the success stories in the book, it would be the HPV story, because the goals of these early researchers who thought that cancer was caused by viruses is really vindicated by all the research done on HPV and cervical cancer and throat cancer and other cancers as well. And it's remarkable now that we have a vaccine that can effectively wipe out some of these cancers if it was widely adopted. The important work was done by a German virologist called Zurhausen, and he was able to show that this strains 16 and 18 initially were the strains that caused HPV-related cancers. and it wasn't other things that people at the time thought that perhaps it was herpes virus. His work was then picked up by others around the world, including Australian researchers who were able to show that HPV virus actually assembles into shells without the nucleic acid in it. So it's sort of these empty little balls, which could be used as a vaccine. There's a race for this that was done between the Australians and the Americans. And eventually, a lot of this was settled in patent court, where it went to court about who owned the necessary intellectual property. And we end up with this glorious case, really, where one form of cancer, we could wipe out this century or in the next few decades. And I see in Australia that they have amazing data now showing that young people just aren't getting cervical cancer at the same rates as their parents did because of the vaccine. The chapter in the book deals with all the controversy at the time and trying to convince people that this was the right approach. It becomes clearer and clearer over time that Sir Hausen was on the right track and that this was real, and there's maybe more strains of HPV, and the virus, the vaccine has gotten better and better over time too. Yeah, I think what's perhaps interesting is just to get to know some of the scientists that uncovered these discoveries and to see how they were motivated. Initially, actually, they had trouble convincing pharmaceutical companies to make a vaccine, but luckily for us, we have one now.
I highly recommend this book. I am someone who has adored the history of science, and more specifically, the history of molecular biology for the majority of my life. And this is a fun read, and it's interesting, and you will learn about the people behind the science that made it work.
Well, thank you, Mark. It took 10 years to write that book, and I tried to make it as accurate as I could. Yeah. And accessible, which is kind of a trade-off that you can have. But
it always is. But I think in my reading of it, you did a wonderful job at it. And I find that if I say I like book by X person, then invariably I say that to someone who doesn't care for that author. So I kind of stay away from that and simply say that this is as good as any of the other books that have really influenced me. And it's the kind of thing, for example, if I were still teaching, as I'm now in my last semester of it, I used to teach first-year seminars where we would actually read deeply and write summaries. I would use this book in the future. I really would, because I think first-year undergraduate students could get most of it, and they would come away with a better view of how science is done.
Well, thank you for your kind words.
Well, it's been a pleasure having you here today, and I want to wish you and yours the very best.
Thank you, Mark. Yeah, thoroughly enjoyed it.
I hope to talk to you soon.
Bye-bye.
This has been Matters Microbial, a podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with, as always, epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Gregory Morgan can be found in the School of Humanities, Arts, and Social Sciences at Stevens Institute of Technology in Hoboken, New Jersey. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.